Hi, today I'm doing my presentation on CRISPR Cas9, and I'm gonna adjust the brightness. Oh my god, I hope you can see, sorry. So what is CRISPR? CRISPR is an acronym, clustered, regularly interspaced, short palindromic repeats. So what CRISPRs are is they're areas in bacterial DNA that actually store DNA from viruses, and it does this in a part of its like immune system. This is how bacteria, yeah, it's immune system basically. So hopefully you can see here, little black diamonds are the bacterial DNA. These little colored ones are all like past DNA collected from viruses in that have attempted to infect it. So it does this using a Cas9, which is a cutting protein. So this big purple thing is the Cas9. So it actually releases strands of DNA. So say this is a past virus that attacked. This is a piece of, RNA, this red, that it matches the DNA sequence of this viral DNA. So the Cas9 will go along. It actually unzips the viral DNA until it finds a matching set with its own RNA here, and then the Cas9 will actually cut this DNA, and when the DNA is cut in half, like a double helix cuts of both strands, it will completely disable it. And then if it was a new one, it would store this DNA in its own DNA. And utilizing it, scientists can actually use it for genetic modification. It's one of the newer ones that's discovered, but it's the cheapest and most effective one so far. Basically what scientists have done to use it is they created a single guide RNA that they select. It's about 20 nucleotides long, and they insert this and the Cas9, which is this big red blob, just into human cells, animal cells, well, usually not human cells, but you know, in a human, but maybe in a petri dish, plant cells, and then it goes along the DNA here of the whatever cell it is, and binds to whatever it needs to, and then Cas9 will do its thing and it will cut the DNA. So it cuts DNA in specific genes. We didn't really go into genes in class, but genes are basically sections of DNA that produce their own proteins, make us who we are. So, you know, this gene, say, makes hemoglobin. There's ones that make keratin for hair, FGF5, which is the hair growth cycle gene. So by cutting the gene, they can actually insert new DNA into the gene. So they just put the DNA in the cell, and when the Cas9 cuts the gene, the new DNA, and it's single strand, not the double helix, a single helix, it will actually fill, it's this green section here is the insertions, the blue is the deletion. It will fill in that space and act as a guide for the DNA to repair itself, and they can either induce a certain mutation this way or get rid of a mutation, depending on the situation. So the first experiment I found for this is hair growth cycles in rabbits. Their aim was to increase hair growth in rabbits for production, whatever they use rabbit fur for. There's the growth phase, transition phase where it attaches, and then the resting phase, and that's when the hair falls out and the cycle continues, and the variation of the cycle depends on how long the hair is, or how long the hair is depends on the cycle, basically. So they mutated this gene and they put it in little rabbit zygotes and implanted it into the rabbits, and when they were born, they did in fact have longer fur. So the blue is the rabbits with the longer fur here, the mutated ones, and then the red is the control. And then even in the pictures you can see it's noticeably longer and thicker. And then the second study was on rice, which is a huge staple food group for much of the world. So they're just looking at increasing crop yield, and they did this by targeting 2 genes, OSFWL4, which is plant tiller, and in fact these red ones, which are the modified ones, for plant tiller is greatly increased, and for yield per plant it's greatly increased. Grain weight decreased, and the number of grains of rice per tiller remained about the same. The second one they chose was OSFWL1, which is grain length. And then the grain length actually increased quite a bit, but the overall weight and size didn't change much. So, not an ideal experiment, but we're still learning a lot about genetics and genes and all that. So it's just showing that they have in fact changed rice. And then in future use, biofuels are a big target, and algae specifically, because it doesn't compete with food crops very much because it's in water, and it grows pretty abundantly on its own. So their aim would just be to grow a optimal strain, basically that can just thrive in any environment and produce maximum amount of biofuel, and it works well because it's kind of like a cycle, because what grows algae is sewage and CO2 and sunlight, which is pretty abundant. And then one of the most obvious ones is human genetic engineering. So we're looking at designer babies, and there's the picture of them choosing their little eye color for the little baby there. This is actually probably the furthest thing from what scientists are looking at achieving with CRISPR Cas9 at the moment. If anything, they're more looking at using it for genetic disease. I saw cystic fibrosis come up a few times for things that they're kind of looking at curing. Well, not curing, but so yeah, basically how that would work is they would want to find the gene that targets cystic fibrosis, cut it, and then insert a new healthy gene. But of course, there's limitations to CRISPR right now. They don't know if CRISPR might unintentionally target a different gene, or go somewhere else, or not target every single DNA strand in every single cell, and then you still might get it. So yeah, so they're still far away from using this on humans, but even if they do in the future, there's still got to be an ethical debate, and then the consent debate, because obviously if you decide to change your baby's appearance or sex or whatever you want to do with it, maybe it grows up and decides it doesn't like whatever change that you specifically made, and no longer it's nature, and now it's oh no, you did this to me. So there's always going to be that. And then there's, if you decide to do it to yourself but you affect the cells that pass on to your children, then your children may get whatever mutation you got, and perhaps it's a good thing, like curing cystic fibrosis, but perhaps there was a bad side effect, and then that's going to be heritable down your entire line. So in conclusion, yeah, it's still new, still being researched, still a long way to go about it before they're going to use it in humans, but they are using it in things like plants and other things to improve quality of life and production and quality and all that kind of stuff. So don't worry, there's not going to be anything crazy happening in the near future. It's just they're just working on it still experimenting. So but I think it's a really cool concept, and it has a lot of potential, and maybe one day in the future it can cure diseases that aren't curable, and just increase quality of life in lots of ways, and provide solutions to things that we didn't know there could be solutions for. Okay, thanks.